Photosynthesis requires many different biochemical pathways and it's surprisingly complex. But before we move on to that, it's good to recap leaf adaptations to aid photosynthesis. The key thing here is it aids photosynthesis. So it's not talking about the leaf structure, it's talking how it helps. So at the top, we have the waxy cuticle, which is transparent to allow light through. You have to explain or understand that all these features are to aid or support photosynthesis. So the light passing through the cuticle will help it strike the chlorophyll, which is below in the palisade mesophyll. The vacuole pushes the chlorophyll to the edges so that it maximizes its light chances. They're also stacked in thylakoids, which we'll look at in a few moments. To deliver the water and the ions and remove glucose, we have the vascular bundle, the xylem and the phloem, vital for the delivery and removal of essential products that are involved in photosynthesis. Below the leaf structure itself, you have the guard cells, which control water loss. They also will sort of prevent or allow diffusion of gases, but the main reason they are there is to control that water loss. And it's the stomata that allows the gases to diffuse in and out. The final structure of the cross section is the airspace, which allows the gases to diffuse directly to all cells or directly away from all cells to allow for maximum photosynthetic and respiration chances. Also, the leaves themselves are structured in such a way that they are broad to maximize the light capturing potential. They are thin to allow light to pass through. It doesn't want to pass fully through, even though it does. It wants to pass through the leaf as deeply as possible so all the chlorophyll can capture light if possible. They are also arranged in stacks so that the light, if it passes through one leaf, will be caught by a leaf further down. But there's also the fact that you don't want to have the leaves blocking each other. They're arranged in a splayed pattern and are non-overlapping, so they do not shadow or block the leaves below. Obviously, there will be leaves below, but they are further below, not directly one on top of another. Just to reinforce this non-overlapping, what I've done is I've drawn a top-down view of a plant like a nettle here. They've got distinct diamond-shaped leaves, and they're arranged in two layered stacks. So we've got leaf one level here, which is pretty much coming in and out of the screen. Unfortunately, I can't draw it 3D, but you get the idea. And below it, you've got leaf two layer here, which is running at right angles to leaf one layer. Then further down, you'll have another set. So from the side, they're capturing light all the way round. As the sun moves across the sky, they can capture it from all locations. And from top down, it's maximizing the chance of light striking the leaf. There's very little gaps between the leaves themselves, so it maximizes potential. Yes, they are at different layers, different stages on the stem, but they really help to maximize it. Also remember the plant will use phototropism to bend towards the light to maximize its light capturing chances. Just before we move on to photosynthesis, we're going to look at the structure of the chloroplast and how things are arranged there. This is in the year one A-level biology, but it's good to recap it. So we've got a nice cross section there, and within the cell of the plant you have the chloroplast, which has got the granum and the thylakoid layers, and surrounding that is the stroma. So it's kind of like an alien city, and you've got the palisade cell moving into the chloroplast through the envelope. Remember, it's a double membrane. We've then got the grana, which is the stacks, and connecting the stacks together are the intergranal lamella, and the thylakoids themselves are the individual layers. I like to think of them as like poker chips or something like that are stacked one on top of the other, and you've got lolly sticks going between them that provide support and structure. They also assist with capturing light. Within the thylakoid membrane is the chlorophyll, and there's also within them DNA and ribosomes, which will assist with the manufacturing of essential proteins. In the stroma surrounding it, you've got enzymes and starch grains which act as an energy store. So any excess glucose could be converted into starch for storage there. We also need to look at a bit of chemistry because this is pure biochemistry pathways. So we need to understand oxidation and reduction. We're not going to go into a huge amount of detail about it for biology, but just the simple thing is it's oil rig, which is oxidation is loss, reduction is gain, and it's of electrons of hydrogen in this case. So that's what we're purely focusing on, the electrons within the hydrogen being either lost or gained. So if oxygen is gained, then energy comes out. If energy is put in, oxygen is lost. So it's really good to sort of bring that in when you're talking about reduction and oxidation, which we're going to look at in a lot more detail in a bit. So photosynthesis is a very complex process. It's split into two stages. The first stage involves the light dependent reaction. it relies on light to work, hence the name. So light strikes the chlorophyll and water is split into component parts, and also electrons are energized due to this. So it converts light energy into chemical energy, which is the ultimate goal of the first stage. What happens is electrons flow through the membrane, water is split by photolysis, and protons, electrons, and oxygen are liberated, which are used for different purposes. Oxygen is actually a waste product which diffuses out of the leaf. The key products that are made at this point are reduced NADP, often referred to as NADPH, ATP, and that waste product, oxygen. What happens in the second stage, the light independent, is the protons, the hydrogen ions, are used to reduce CO2 and convert it into glucose ultimately. We'll look at that when we look at the light independent stage. Chlorophyll is a very complex molecule. Luckily, we don't really need to know much about the structure, and it's very similar in its structure to hemoglobin. But instead of having iron in the center, an ion of iron, it's got the magnesium there. That's why magnesium is vital for chlorophyll structure and also vital for photosynthesis. Magnesium deficiency can have detrimental effects to plants. This is one of the effects of acid rain. It actually leaches the magnesium out of it, so it loses its chlorophyll. There's also different types of chlorophyll A, chlorophyll B. You can look those up in your own time. They're not needed fully for the course, but an awareness of them is always good. Key terminology is very important here. And when we're looking at the electrons being excited, we're actually saying they have been photoionized. So the act of photoionization makes an ion of the molecule that the light is striking. So it's literally electromagnetic radiation striking a molecule and releasing an electron or exciting an electron. So it gains so much energy, it escapes from the molecule. In this case, from the chlorophyll. So the light strikes the chlorophyll and the electron is given energy and raised to a higher energy level and literally is lost by the chlorophyll. That then can impart that energy to other processes, such as the moving of molecules across the membrane. It also gives off heat and sometimes light as well. So we've got this multiple effects. So light excites an electron, which is then released into the surrounding tissue, and that excited electron can impart its energy to other things. It acts as an energy donor, for want of a better term. The key thing of this whole process of light dependent reaction is to make high energy units. We start with sunlight, ADP and phosphate ions, spent energy, water and oxidized NADP, a relatively useless chemical at the moment because we need to reduce it so it can provide reducing power to help in the next cycle. So it starts with those and it ends with ATP, high energy unit, oxygen, waste product, and that reduced NADP, which works as a reducing agent. That is vital to convert carbon dioxide into glucose. Before we look at the in-depth process, we do the light-dependent reaction in a lighter version. So what happens is light strikes chlorophyll, an electron is excited and photoionized. That then escapes the chlorophyll and is captured by a carrier in the thylakoid membrane. As it moves through that membrane, it's going to impart energy for a particular process. That is an oxidation step of the chlorophyll itself. The electron then moves through the carriers, which we'll look at in more detail in a bit, and imparts energy ultimately, not directly, but ultimately it's going to impart energy to allow the manufacture of ATP from ADP and a phosphate ion. So they will combine together to make ATP. It's not a simple step as it's shown here. This is just the real light version of it. We're going to leave that electron there because at the same time light is striking the chlorophyll, it also strikes water and splits it into an electron, a hydrogen ion, and oxygen, the waste product that diffuses out of the leaf. That electron will be used to return to the chlorophyll to stabilize it. So we are now reducing the chlorophyll to return it back to where it should be. It's basically lost an electron, it's now gained an electron, it's back to a happy state, for want of a better term. We've now got that hydrogen ion floating around, the electron held in the carrier there, and we've got that NADP, which is currently oxidized. We need to reduce it. So what we do is we take the hydrogen and the electron, combine it with the NADP, and we end up with reduced NADP, sometimes referred to as NADPH. We have now made our high energy ATP and our reducing agent, which can go off to the light independent reaction to, for want of a better term, power that. Obviously, this all happens very fast and simultaneously. So light doesn't strike one bit then another, it happens at the same time. So I've tried to animate it in one go there. It's not the best thing, but at least you can see that it's all happening kind of in a step-by-step process, but simultaneously. So that's what actually happens. And we've got two things we want to look at. There is photosystem 1, which is this area here effectively, which we'll look at later on, and photosystem 2, which is the first bit. Just to confuse the issue, photosystem 2 happens before photosystem 1. I'll explain about that later.